Okay, everyone, can you hear me? Today we will talk about seed germination, and hopefully we will finish the lesson today. If not, we will finish it tomorrow in class. Okay, this is a section that we did not discuss in class, which is seed germination. Okay, can everyone hear me? Okay, can you hear me? Is my voice clear?
Yes.
Yes.
Okay, we'll start seed germination. This is mainly practical, mainly paper 6. Now we need to explain what is seed germination, what happens in seed germination. Seed,
لا ودي بتبدأ 59 مش بتبدأ واحدة.
excuse me? Seed germination, this is after all the steps that we talked about in class. We were talking about plant reproduction, and if you remember, we were talking about sexual reproduction in plants. Quick revision to sexual reproduction in plants. When we want to study sexual reproduction in plants, we always start with the flower, because the flower is the reproduction. Okay, don't bother about recording the session. I will record it for you. The flower is the reproductive organ in a plant. We will draw the different parts of the flower. We will draw the female and the male parts of the flower. If you have a question, ask a question. We said that this is the female part. What is this part called? Who will remind me?
Stigma.
Very good. Followed by the style. We are revising.
And then the ovary.
Okay, yes, and then the ovary. What is found inside the ovary? What are these structures that I am drawing? The
ovules.
ovules. And please, in plants, the female egg is called the ovule. In humans, it's called the ovum or the egg. So don't write egg when you're talking about plants, okay? Now, this is the female part, which is called the carpel or the pistil. And then you have the male part of the flower, which is made up of two parts only, the filament and the what?
Anther.
Very good, the anther. And the anther, what does it produce? The pollen, okay? So if you are given a picture like this and you are asked, where does meiosis take place? Give me two organs where meiosis takes place. What will you say?
Stigma.
In the ovary.
In the ovary. Who said stigma? Meiosis does not take place in the stigma. Meiosis takes place in the ovary because the meiosis, its function is to produce the ovule. So meiosis takes place here in the ovary, and the meiosis also takes place in the anther to produce the pollen. Okay, meiosis takes place in the anther and in the ovary. Fine. Now, after the pollen is produced, the pollen needs to fall on the stigma. How does it fall on the stigma? The pollen is carried to the stigma by one of two methods. Who will remind me?
Insects.
Very good. Either by insects. Now my picture is gone. Either by insects
or
wind. Very good. It's like a story. If you understand the story and study it from the beginning till the end, it will be very easy. The pollen will fall on the stigma. After it falls on the stigma, what will grow down the style?
Pollen tube.
A pollen tube.
Very good. And what is carried down the pollen tube?
The pollen.
The male nucleus. Yes, you are not wrong, but please mention the term male nucleus. You will always find it in marks. Okay, the male nucleus, because this male nucleus will then enter the ovule, and the male nucleus and the female nucleus will fuse, and this process is called what? Fertilization. Okay. Now, when all the ovules are fertilized, these ovules will become what? Seeds.
The seeds and the ovary will become the fruit.
Very good, excellent. This will become the fruit. After all the ovules are fertilized, all the different parts of the flower will fall off because we don't need them anymore. The stigma, the style, the stamen, the filament, the petals, everything will fall off except the what? At the base. The
petals?
Not the petals, no, the petals, the sepals. The sepals, these are the small green-like structures. They look like petals, but they're smaller at the base of the fruit. and they used to protect the flower when it was in a bud. If you want to remember how it looks like, draw a strawberry. This is a strawberry, and the base of the strawberry is something green here. This is the sepal that remains after all the parts of the flower fall off. So this is the story of the fertilization of the ovules and the making of the, what's this called, of the seeds and the fruits. Now the seed, this seed will germinate when it falls on soil and water. It will germinate. Germination is what you see here right in the picture. This is germination. This is the growth of a new plant from a seed. So we will study the different parts of the seed and how does germination happen. This is germination here, right? You see here in the picture, you have a seed. The seed will grow a small protrusion down there called the radicle, and then it will grow a shoot and it will grow into a plant. Now, what are the different parts of the seed? You need to know the different parts of the seed. There is an outer coat here called the testa, and this testa is hard. And what do you think is the function of the testa? This outer coating of the seed. What do you think?
Protection.
Very good. Very simple function, protection. It will protect the seed. Okay. Now, inside the seed, let's look inside the seed. There is a small baby plant. And this small baby plant is divided into two parts. It's divided into something called the plumule and the radicule. The plumule and the radicule. This is the embryo plant, the baby plant. The plumule will grow to be the future shoot and leaves. And the radicule, it starts with the letter R, and it will grow to be the future root. Okay, so you have a baby plant inside the seed that is divided into two parts. The first part is called the plumule, which will grow into the shoot and the leaves. And then you have the radicule that starts with the letter R, and this will grow to be the future root. Okay, and by the way, for your information, the radicule comes out first. So the radicule appears first and then the plumule. And there is a very small hole. This hole is very important in the seed. This hole is very small, you don't see it. It's called the micropyle. What is it called? The micropyle. Now, the micropyle has two functions. Function number one, it allows the entrance of the pollen tube during fertilization. Function number two, it allows the entrance of water during germination. So when will you mention the micropyle first when you're talking about the germination of the pollen tube? Let's go back to my flower. This is my flower. I'm repeating several times here so that you don't lose track. This is the female part. This is the filament. This is the anther. The pollen will fall on the stigma either by the wind or by the insects, and then the pollen will germinate into a pollen tube. A pollen tube will grow down the style. The male nucleus will move down and down and down and down until it enters the ovule. How does it enter the ovule? Through an opening known as the micropyle. So when you are asked to discuss the process of fertilization, you have to mention all those steps, including the micropyle. Okay? So these are the different parts of the seed. Very easy, the different parts of the seed. How will I draw the different parts of the seed while I am studying? I'm going to draw something similar to a circle. Now help me label this outer coating. What is it called? Testa, for protection, okay? Can you please mute your sounds? I have a small baby plant here that is made up of two parts. Whatever will grow into the future shoot and leaves is called the plumule. Memorize with me, plumule. This is a U. And then whatever will grow into the future root is called the radicule. Radicule. And the radicule always appears first. There's a small opening here called the what? Micropyle, to allow the entrance of the water in germination and allow the entrance of the pollen tube in fertilization. Now, I have here lots of space inside the seed, all this. What is all that? All this is called the cotyledon. The cotyledon is actually stored food, mainly starch. Okay, so this cotyledon is the stored food, mainly starch. And why do you think it is important? What does it give the baby plant? Someone give me an answer. A very easy question. These cotyledons, the stored food, what does it give the baby plant? What does it give the baby? What does the baby plant need to grow?
Energy.
Exactly.
Energy.
Energy. Very simple. Energy. Okay. So let's go back to the pictures. Here is also the ovary and the ovule inside it. And this clearly shows you the opening. We're talking about the micropyle that allows the entrance of the pollen tube and water. And here is germination. You start with a seed, and the first thing that appears from the seed is the radicule, okay? And then the rest of the structures. Here you have, again, the structure of a seed, the testa, the coat. You have the baby embryo, the plumule, that will be the future shoot, or the leaves, and then you have the radicule that appears first, and all this are storage cotyledons, stored food, because this stored food will be broken down for germination to start. Now, I have a question for all of you. How is the food broken down to give energy? What is the name of the process that breaks down food to give me energy?
Digestion.
Digestion.
Digestion in your body. Yes, digestion in humans, but in plants, do they have a digestive system?
Photosynthesis.
Photosynthesis.
No, it's not photosynthesis. Again,
Firing.
Respiration.
now we have to understand the difference between photosynthesis and respiration. Let me open the screen. Okay, give me a second. Now, photosynthesis and respiration, don't worry, we will study photosynthesis in detail, but we need to revise it now. Respiration, this is a process in which food is broken down to give us energy. Respiration is a chemical reaction that takes place in all living things, all living cells. If you remember, it's one of the characteristics of living organisms. Anything alive does respiration. Respiration here is when you have food. Today or now, we will study only the word equation, and then we will study the balanced chemical equation when we take the lesson respiration. Food is broken down in the presence of oxygen to give me what? Carbon dioxide and water. This respiration, this reaction takes place in your body, in the body of plants, in bacteria, in anything that is living. Plants, they do something extra. Plants, they do, or they respire, plus they do photosynthesis. What is photosynthesis in plants? Photosynthesis, this is when plants make their own food. It's actually the opposite of respiration. Plants, this is a plant here. It will take water from the soil, and it will take carbon dioxide from the atmosphere, and it will combine the water with the carbon dioxide to make food and make oxygen. So photosynthesis is actually the opposite of respiration. Photosynthesis in plants is water from the soil plus carbon dioxide from the air to give us glucose, C6H12O6, to give us food, which is glucose, plus oxygen. And to balance that, we add 6, 6, 6. So photosynthesis, this is the process used by plants to make food. When the plant is still a seed, and this seed grows into a new baby plant, it first starts by the process of respiration. Why does it start with the process of respiration? Because it already has stored food. It will first break down this food and grow a little bit, and when the food is finished, it will start to do photosynthesis. Again, I am now studying germination. Germination is the growth of a baby plant from a seed. I have a seed, and this seed has certain structures. It has an outer coating called testa. It has the baby plant here waiting to grow. It has an opening called a micropyle, and it has lots of stored food called cotyledons. The plant, or this embryo plant, wants to grow. How does it grow? First, I want you to understand something. This seed is below soil level. This is soil. It is underground. So is this seed exposed to light? Is it exposed to light?
No.
No. So how can it photosynthesize? It cannot photosynthesize yet because light is essential for photosynthesis. It cannot photosynthesize. So this baby embryo plant starts growing by first starting respiration. It does not photosynthesize. for several reasons. Number one, because there is no light underground and it needs light to do photosynthesis. Number two, it already has some stored food. It will take this stored food from the cotyledons and break it down in respiration to give it some energy, and this energy will help it grow. So what does this baby plant do, this embryo? It will take the food, break it down, plus oxygen, to break it down in the presence of oxygen, because this is aerobic respiration, to give me carbon dioxide and water. And of course, this provides energy. This energy will be used by the plant to grow into a small baby plant, and this is germination. Clear? So germination starts by which process? Respiration or photosynthesis?
Respiration.
Respiration. Again, we are talking about germination in plants. Germination is after the ovules and the ovary have been fertilized and the fruit now has seeds, and this seed is put in the soil. I have a seed and it is inside the soil. This seed will grow into a baby plant. Plants, they do two things the whole time. They do photosynthesis, respiration, photosynthesis, respiration. You all know that photosynthesis needs an essential requirement. What does it need?
Light.
Light. When this seed is underground, when it is in the soil, do we have any light? No. So this seed will not photosynthesize, not now, not yet. Then how did it start germination? By respiring first. How does it respire? Because it does have a reserve of stored food in the cotyledons. It will take the reserved food and break it down in respiration. It will give itself some energy. This energy will be used by the embryo plant to grow. Once it appears above the soil and it is exposed to light and all its food reserves are finished, only then will it start what? Photosynthesis. Okay? Now, why is this important to understand? Because sometimes they will give you a graph. This graph has time plotted on the x-axis and the dry mass of the seed on the y-axis. And I will discuss the term dry mass in a minute. This is the weight of the seed, the mass of the seed. The mass of the seed, as it germinates, it will drop first and then it will increase. And they ask you, why does the mass of the seed drop first before it starts to increase? Why does it drop as it starts to germinate?
Because food is being used up for energy.
Excellent. Because the stored food is being broken down. Because we start germination by respiration. So here, when the mass of the seed is falling, the seed is respiring, and the stored food or the cotyledons are being broken down. And then when the dry mass of the seed increases, this is when the seed will start to do what? Respiration or photosynthesis? Photosynthesis. This is when it will start to make its own food, and the food will increase the mass of the seed. Who has any questions in what I said? I'll repeat again for those who were not paying attention.
Doctor, can you repeat the part?
Yeah, which part? I'll repeat all of it. Be patient, okay? I'll repeat everything. We're talking about the seed. This seed, you need to discuss or talk about its structures, the inside of the seed. What's very important is that you have a baby plant, and the remaining part of the seed, most of the seed actually is stored food. This yellow part is stored food called the cotyledons. This seed is below the soil. It is not exposed to light. Even though it is below the soil and it is not exposed to light, it will grow into a baby plant. So it cannot start growing by photosynthesis because photosynthesis needs light. So what did this seed do in order to germinate? It started the germination process by respiration. In other words, it took this stored food was broken down. It is broken down in aerobic respiration to give me carbon dioxide and water and energy. It used this energy to grow. That is why the mass of the seed at the beginning of germination drops before it starts increasing again. Who is still confused? Okay, let us look at pictures. As long as I'm not receiving any questions, so I expect you to understand what I'm saying. This is the structure of the seed. I hope it's clear. And please don't forget the micropeel. The micropeel is important. Okay, fine. Now let's look at this table. You have it in your notes, which tells you the function of every part. Testa, of course, this is the outer covering for protection. The cotyledon, this is the stored food. It will be used in seed germination. Sometimes you have two cotyledons. In this case, the plant will be called a dicot. An embryo plant, this is the baby plant that is made up of the radicule and the plumule. The radicule will grow into the root and it appears first, and the plumule will grow into a shoot. If you have any questions, stop me, okay? The micropeel, this is the small hole that allows the entrance of the water for germination, and the pollen tube. Okay, I don't think anyone has any questions. And pictures and pictures and more pictures. Now I want to go directly to the graph that I was talking about. Here is the graph that I'm talking about. Yes, Nariman? You have a
Can you
question?
please repeat?
Okay, I am repeating. Here is the graph that we were talking about. This graph, what is it showing you? It is showing you the time on the x-axis and the mass of the seed on the y-axis. Nariman, the seed, when it starts to germinate, its weight will decrease, its mass will decrease or increase. What do you see on the graph? You see the mass of the seed decreasing?
It will decrease.
Excuse me?
Yes, it will decrease.
Yes, very good. The mass of the seed will decrease. Now the question is, why does it start by a decrease in mass?
Because it stores food?
Because it stores food. Yes, it stores food. Now, what does it do to this stored food? This stored food was used to what?
Respiration.
In respiration, correct? Look here, this is the seed. This seed, it did not germinate yet. Its mass was high. And then it will start to germinate. As it starts to germinate, the mass falls. Why? Because the seed is underground. It is not exposed to light. It is not doing photosynthesis yet. Then what does it do? It is doing respiration. What is respiration? It will break down the stored food so that it provides itself with energy, and then it will use this energy to germinate, as you can see. Once it germinates and it appears above the soil and now it is exposed to light, it will start doing photosynthesis, and only then will the mass of the seed increase. Here the weight will increase as the seedling can do photosynthesis and grow. Yes, Hadiya, you have a question?
Yes. It's breaking down the food, which
Yeah,
is decreasing the mass, right?
okay.
But at the same time, it's also growing, right?
Okay, it is growing, but the stored food, it weighs more than the leaf that is erupting. The leaf is much more lighter. So it is losing weight. It is losing in mass because all the stored food is breaking down. You have to break it down first in order to grow. You don't grow and then break down the food. You have to break down the food first to give you energy and then grow.
Okay, thank you.
Okay?
Doctor, when does the seed start to photosynthesize?
Very good question. When it starts to see light, because photosynthesis cannot happen without light. Okay?
Thank you.
Very good. This was a very nice question. Now, this graph, if it comes to you, it will come in paper 6, and then they will ask you this classic question that they always ask you, describe the graph. When you describe the graph, you always have to take numbers from the graph. We know this by now. So you will go to the graph and then you will use the days and you will describe what is happening and you will explain. This is the part between description and explanation. So the describing part, from day 0 to day 9, the dry mass will decrease as the seed germinates. Why? Because the seed will start the process of aerobic respiration, using up the food stores in the cotyledon. So this is the description and the explanation. And then from day 10 to day 30, here, from day 10 to day 30, let's assume, this is the part where the mass will start to increase. The dry mass will increase because now it will start doing what? Photosynthesis, and start producing what? Producing food. So the mass will increase. And then from day 30 to day 40, this graph normally continues. It will drop again because the plant here, this is the life cycle ending for the plant, and this when the plant dies. Okay? This graph, is it clear? Again, here,
Yes, excuse me.
yes.
Is the number of days on the graph, are they set numbers? Like, should
No,
we
they're not set
move-
numbers. It differs
They're
from
not
one
like the
graph to the
menstrual
other.
cycle, no?
No, they're not set numbers. No, they differ from graph to graph.
Okay.
Okay. Now, how do I erase whatever I have here? I actually forgot. Clear or drawings. Give me a second. Yeah. Okay. Farah, do you have a question? Farah, your hand is raised. You have a question?
No, ma'am.
Okay. Now, I don't want to jump. I actually did. So we will go back to the beginning of the lesson so that you study it in the order in your notes. The definition for germination, this is the process by which an organism grows from a seed, a new baby plant growing from a seed. Very easy. Now, there's a term here that we need to understand. You have seeds now as we are speaking in the kitchen, in your kitchen at home. And these seeds are alive. They're not dead. But why do these seeds do not germinate? In your kitchen now, if you go to your kitchen, you will find popcorn and you will find lentils and you will find fool and you will find, these are seeds. If you take those seeds and you put them in soil and you give them some water, will they grow or won't they grow? What do you think?
They
If
will.
they grow. Okay, why did they grow when you put them in soil and water and they're not growing in your kitchen shelf?
Because they have the suitable conditions when they're in the soil.
Excellent.
They need energy.
Because they need what? Because someone...
Energy and minerals to
Yes, you are correct. Okay, when I have a seed, I like to draw. When I have a seed in my kitchen shelf, this seed, it will not germinate. It will stay dormant. You need to know this term. It will stay dormant or inactive. In other words, it is sleeping, but don't write sleeping on the exam. It is inactive or it is dormant. Why? Because it is not surrounded by the suitable conditions, like your friend said. Because this seed, in order to germinate, it needs, number one, water. Number two, a suitable temperature. Number three, it needs oxygen. All these things must be present for germination to happen. That is why when I took the seed and I put it in the soil and I started giving it water, only then did it start to germinate. Okay? Now, water is needed for many things in germination. There is a list that you need to know. Water is used. First, water is absorbed through the micropyle. I think you know what the micropyle is now. That's the small hole in the seed. When the water enters into the seed, this water is needed, number one, for the activation of enzymes in the seed. Any process starts or is catalyzed by enzymes, photosynthesis, respiration, we need the enzymes. So the water is needed to activate the enzymes. Number two, the water is needed to convert the stored starch into simple sugars so that these sugars can be broken down in respiration. Number three, the water is needed to transport sugars and the amino acids within the seed. The water is needed for the vacuoles inside the plant cells to start expanding and growing. The water is needed to increase the water pressure inside the seed, which is, you know by now, called the turgid or the turgor pressure. Water is needed in photosynthesis once the plant becomes exposed to light. Okay, so this is a very, very common question, and we will revise the uses of water when we study plants again. Water is very important for living organisms. We studied water and its importance in our bodies. Now we want to study water and germination. For a seed to germinate, you have to give it water. It will not germinate without water. When it is in your kitchen cabinet, it is dormant or inactive, and it is also described as dehydrated. What is dehydration? Dehydration, it has water or no water?
no water.
No water, okay? So this seed, I don't know if you did it in year 5 when you were little. You took seeds and you put it on wet cotton. Did you do this experiment in class? You put it on wet cotton, not even in soil, and it starts to germinate. Anything that is wet will make it germinate. So the water is needed for these reasons. Please memorize those 6 points. activation of enzymes to dissolve the stored food, to transport the food around, for the expansion of the vacuoles to increase the turgid pressure, so the vacuoles will start expanding, and then when the plant is exposed to sunlight, to start photosynthesis. Okay? Now, the second requirement for germination, oxygen. Oxygen is needed for what? For what?
Aerobic respiration.
For aerobic respiration, because we said that the seeds, in order to germinate, we start by aerobic respiration. We break down the food and the stored cotyledons with the help of oxygen to give us energy so that we can grow. So oxygen is needed for germination, for aerobic respiration. That's the only use for oxygen. And then we need to give those seeds a suitable temperature. If those seeds are in the freezer or in the fridge, will they grow? Give me an answer. If I put those seeds,
No.
no, because the enzymes, are they denatured or they are inactive?
Inactive.
Inactive, of
Inactive.
course. The enzymes, they slow down, they are completely inactive, but they're not dead. The temperature has to be nice and warm and suitable. Why? For the what?
For the enzymes to work.
For the enzymes to work, because enzymes, as you know, they have an optimum temperature. In plants, the optimum temperature for the enzymes is different than the optimum temperature for your enzymes in your body. In humans, our optimum temperature is 37. In plants, it's nearly 40, 41. Okay? So you need to put the seed in a nice warm environment that contains water and oxygen for it to germinate. Now, these are the requirements for germination: water, oxygen, and warmth. Warmth means warm temperature. So this is how we will memorize the requirements of germination. Whoa. The first W is for water, and the O is for what?
Oxygen.
Oxygen. And the last W is for
Warmth.
warmth.
Or
Okay. Okay, did I mention light here? Was light needed for germination?
No.
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 Light is not needed for germination, because germination, we said, starts by which process? Respiration or photosynthesis?
Photosynthesis.
Inspiration.
Respiration.
Respiration. So first, the seed is in the soil, no light. So the seed will grow even though there's no light. Why? Because the seed will break down the stored food in respiration to grow a small baby plant. Once it reaches above the soil and it is exposed to light, only then will it start photosynthesis. So for germination, light was not needed. What did we need? We needed water, oxygen, and a suitable temperature, which is warmth. Okay, so light is not needed, but the light should be suitable also, not extremes of light, but light is not needed for germination. Okay, any questions? Anything in what I said is hard? Okay, as long as I don't have any questions, then I will proceed. Okay, so clearly here, light is not needed for germination, but very high or very low light intensities does not allow the enzymes to function normally, because at some point, the plant will be exposed to the light and I will start photosynthesis, so I will need light then. Okay, now, these questions, they usually appear in paper 6, the germination and the growth of the plant. Now, first, students, when they come to answer questions related to investigations concerning germination, let's say we want to study the effect of temperature on germination. What is the independent variable? If I want to study the effect of temperature on germination.
Temperature change.
Temperature
change.
I want you to be a little bit creative. Don't tell me, because I see very funny answers, I'm going to put the seed in a water bath and boil the seed. When I come to change the temperature in any investigation, there are 2 methods. Either I use a water bath, yes, or what do I use? An oven, an incubator. When do I use a water bath and when do I use an oven? I will use a water bath if I have a liquid, juice, solution, liquid. I will put this test tube that contains the liquid in the water bath. This is when I will use a water bath. But if I have a seed, I will not put the seed in the water and boil the seed. I will put the seed on a petri dish, on a small plate. These are my seeds here. Okay. on, let's say, wet cotton, and I will put it in an oven to change its surrounding temperature. Okay? This is one thing. Number two, when seeds germinate, also in the answer, you will tell me, I will wait for 5 minutes and I will see if the seed germinates or not. Will a seed ever germinate in 5 minutes?
No.
No. At least you give it a week. So be realistic in your answer. You will give the seed a week or 10 days. In other words, for example, if you are studying the effect of temperature on the growth of seeds, then we all agree that the independent variable will be the temperature. And the dependent variable will be what, for example? What will be my results? What are you looking for?
Length of the root.
Yes, you are correct. The length of the part of the seed that germinated, or simply speaking, or easier, the number of seeds that germinated. I will just count them. Or I can measure the length. Okay? So my steps, what do? I will get several Petri dishes, at least 3, and I will put the same number and the type of seed on the Petri dish. I'm drawing it for you so that you imagine with me. I'll put, let's say, 3 seeds. I'll put a little bit of water in every Petri dish, the same amount of water, and I will put every Petri dish in a different oven, in a different incubator. And give examples. One oven will be at 80 degrees Celsius. Another oven will be at 20 degrees. A third oven, let's say, at 50 degrees. And a 6th oven will be at, let's say, 40 degrees Celsius. Can someone give me an idea for a control experiment?
Room temperature.
No, we never use room temperature as a control. We said the room temperature can actually be one of the temperatures. What if your room temperature is 20? You're in the winter and the temperature is 20 degrees. So the control cannot be the room temperature. Give me
The temperature of the seed.
Excuse me?
The optimum temperature of the seed.
These are my results. What will be my control?
Boil.
Boil the seed. I'll boil it. Okay? And I'm going to put it in a Petri dish aside to compare. Fine, I'm going to destroy it. I will destroy the enzymes in the seed and see if it's going to germinate or not. Of course, it will not germinate. And by the way, you don't have to write a control for every experiment. You don't lose marks if you don't write a control. Okay, I'm going to leave those Petri dishes in the oven for a week, at least a week. You all tell me I will leave it for 5 minutes. It will not germinate. None of them will germinate in 5 minutes, even if they are put in the optimum temperature. After the week, I'm going to get out the Petri dishes and I will count the number of seeds that germinated, or I will measure the length of the radicule that appears. Counting the number of seeds will be easier. Okay? And the Petri dishes that shows the maximum number of seeds germination will be the optimum temperature. I'm finished. Easy or not easy? Who has any questions?
Easy.
Okay. Now, I also want to point on something in investigation equations. Do not give me results because some of you will improvise. They will tell me, for example, when the temperature was 80 degrees, 1 seed germinated. When the temperature was 20, none of the seeds germinated. When the temperature was 50, 2 seeds germinated. And when the temperature was 40, 3 seeds germinated. Who told you that? Did you do the investigation yourself? No, you didn't. In the question, you are asked to describe the steps. How will you do it? But don't give results. You can give a guess, okay? Or you can give a conclusion. If the number of seeds that germinated were the maximum, then this temperature is the optimum temperature. This is how you will conclude. This is how you will find out which temperature is the optimum temperature. Okay? But don't give me results because you did not do the investigation. How do you know? Okay, you're guessing only. Okay, going back to germination. Actually, this is a very important part because this always pops up in paper 6. And I want to focus on a term known as the dry mass. Can you guess what is the dry mass?
Mass with no water.
It's very easy. It's very clear. The dry mass is the mass of the organism with no water. And we always use dry mass when we come to measure the mass in plants, because the amount of water absorbed by plants is variable throughout the day even. Plants in the morning, for example, when it is very sunny, absorb lots of water. And at night, when they are not photosynthesizing, they absorb little water. So the wet mass or the fresh mass is the mass with water. The dry mass is the mass without water. Which do you think is more accurate? Dry mass or fresh mass?
Dry mass.
The dry mass. We always use the dry mass when we are measuring the mass of plants, because this is much more accurate, because the amount of water that is absorbed by the plant is very variable. How do we obtain the dry mass? I don't think you will ever be asked this question, but in case, they take the plant and they put it in an oven. In an oven at very, very low temperature overnight, so that all the water evaporates and what remains is the body of the plant without water. And this will be the dry mass of the plant. Okay? All this is written here. This is all practical. Paper 6. Dry mass. Very easy. You don't need to memorize anything. I want you to understand. Dry mass is the mass of the plant after it has been dehydrated, after we removed the water. This is also only used for plants in plant experiments, because it is more accurate, because plants absorb water in variable amounts of water during the day. So if we use the wet mass, it will not be accurate. Okay? Do you understand what's dry mass? And why we use dry mass in plants instead of fresh mass? Any questions?
No, Miss, why do we use dry mass?
Okay, again, why do we use dry mass? Who will answer him? We have 2 plants, my dear. Is this Ali?
لا.
Okay, it's no problem. I have 2 plants. This is a plant and this is a plant. I'm using this plant in my experiments. I want to see how the mass of this plant will change. Okay, this plant A and plant B. Let's look at plant A. If I measure the mass of plant A at 7 a.m. in the morning, it will be, for example, 1 kilogram. If I measure the mass of the same plant at 8 a.m., it will be 1.5